Hej och varmt välkomna tillbaka till Erik Pensers Banks Temadag, Imaging och Diagnostik. And I will now switch to English because we will have Fluoguide with CEO Morten Albrechtsen. Go for it, Morten.
Thank you, Ludwig. Thank you very much for the invitation and introduction, and welcome everybody. I would like to give you a run-through of Fluoguide and what we do. We maximizing surgical outcome by intelligent targeting. If we have the next slide, just our disclaimer, and you should make your own assessment. And have the next again, Ludwig. Next, yeah, thank you. Then really what we do is that we start out with the oncology surgery, and that's because it's a large unmet medical need. Every year, about 15 million patients will have cancer, and about 80% of them will need surgery sometimes during their disease. And the problem is that cancer recur locally in about half of the patient, and it's not because the surgeon are poor in any way, it's just because it's very difficult to see where the cancer stops and where the normal tissue starts. And really our solution to it is very simple. We light up the cancer with a UPAR targeted illumination, and we help the surgeon to remove all cancer the first time. Our lead product, FG1, is in clinical development now. It's for an aggressive brain tumor or cancer. It's a glioblastoma. And it's already now been demonstrated that it lights up the human cancer and also that it's safe. We have a strong basis. We have a strong IP protection. We had good cash position. We just had a directed issue last week. We're listed on Nasdaq First North and have market cap about 1 billion SEK. And then we have a seasoned team that are cross-functional and have a clear plan. And this is what I will, if may have the next slide, that's what I want to tell about. So our procedure is really very simple, and yet it had quite a profound impact. What we do is that our product is injected into the system into the veins of the patient before surgery. It will then circulate in the body, and it will bind to cancer cells. And then during a while, all the product that is not bind to anything will be removed from the body, and only the cancer-bound FG1 will remain in the body. And then during surgery, the surgeon switch on the fluorescent light, and that's a night sight vision. And then they will actually see the cancer lights up, and they can remove quite precisely the cancer, and that's really what we do. And one of the things, there's 3 things that are really important with Fluoguide and makes it a very interesting company, is that everybody likes us as the first one. And that, of course, goes for the patient, which is obvious because we help them being cured. Potentially, at least, we reduce their additional treatments, additional surgery, and more aggressive treatments. But also the hospital, they save money because we save them surgery, we save them more treatment. The surgeons, we make them better to what they really want to do, and that is cure the patient. And then last but not least, the equipment manufacturers, they are also quite pleased with the Fluoguide. And the main reason for that is that we can get features out of the equipment that are not able to get out of the equipment without our products. And that means that the equipment manufacturers either can get a higher penetration of their equipment into the market, or they can sell it at a higher price. So really everybody wants us to see us succeed. And if we have the next slide. So UPAR, what is that? So UPAR is an enzyme system that is used by the cancer to invade into normal tissue. So it's placed on the front of the cells in the front of the cancer, and it's used for the cancer to tear down or break down normal tissue, so it can invade into it and get really metastasize and spread around the body. And that's why UPAR is particularly expressed on very malignant cancer, and also very much expressed on the forefront of the cancer where it needs to invade into the normal tissue. So it's a quite nice target for what we want to do, because it's really placed on just exactly the location where the surgeon needs it, because they can see the middle of the cancer. I mean, that's quite clear. But what really is difficult is to see the forefront and the surrounding of the cancer. And that's exactly where UPAR is particularly expressed. There's also a long and a large body of literature on UPAR. It's been used for drug targeting purposes, used for diagnostic purposes, and a couple of companies come out of this research over time. But what we do at Fluoguide is that we use UPAR to target the precise operation of cancer. So Before we went into human testing, we have done a range of different preclinical models where we have taken human cancer and implanted it into an animal and then make the surgery. And we have proved that it works across many different indications. And of course, some of them are the glioblastoma, which we are working on now. If you look at the image on the right, you can see that it's very precisely, FG1 binds very precisely to where the cancer is and make a very sharp delineation between the cancer and the normal tissue. And that's exactly what we want. We want something that is very specific to the cancer and market, and that's really what UPAR does. So if I have the next slide, please. So this is one example of a study we did. We took a human surgery, we took a human cancer, it was pancreatic cancer implanted into the mice, and we took human equipment. And then we asked the surgeon if they could remove all the cancer in the animals and let us know when it was finished. And after 4 animals, they done under white light where they could not see our product light up, they said, well, now we have removed all the cancer, we've finished, we will send the rest of the patient home. And then we switched it on the fluorescent light, and then the surgeon found cancer in 4 more of the animals. And that really means that had it been patient, those 4 animals, if it had been patient, have been sent home and would have come back either with a regrowth of the tumor, or and then they should have more aggressive treatment, they have less quality of life, and also ultimately they could risk of being dying of it, of course. So in half of the cases, we actually got more cancer removed by our technology. One thing that was quite important is that we used standard equipment. And that is one thing that is very important for us, is that across all what we do is that we want to have our technology implemented easily into the surgical rooms. And that's why we need them to have, or we need to fit into the equipment they already have. So we don't have to ask them for purchase more equipment. So we use standard equipment here as well. It may not be the best of the equipment, but we use what is available out there. And if I may have the next slide. So this is an overview of the market side. It's more than 5 billion euro market that we potentially target with our current plan. And that's because UPAR is, as I mentioned before, as you can imagine, is extensively expressed on most solid tumors because they're aggressive. And that's, for instance, lung cancer, breast cancer, and colorectal cancer. It's not so much expressed on prostate cancer because prostate in generally are not that malignant, and only for the prostate cancer that are very malignant, UPAR will be expressed. So we targeted the 3 first I mentioned, breast, lung, and colorectal cancer. And then there's a couple of indications that are less frequent, but they're still very severe. They have, on the other hand, an orphan drug potential, and one of them, the one we are focusing on, is aggressive brain cancer. One thing I should say is that surgery is used as a cornerstone in all treatment of cancer, and the number of surgical procedures is expected to grow over the time, and that's because, surgery treatment, chemotherapy and radiotherapy is kind of all used for treating the patient, but one of them is often chosen first. And the earlier and more localized the cancer is when it's discovered, the more likely is that surgery will be the first treatment modality to be applied. And that's, for instance, the case in glioblastoma, but it's also the case, for instance, when you have the number of screening programs going on, where you will detect cancer earlier and you will have a higher likelihood that when you find it, it's more localized, and therefore surgery becomes most important. So surgery will at least not be less important as we go on. It will, on the contrary, probably be more important as we go on, and that's actually a very nice trend for us as well. So if I may have the next slide, please. So glioblastoma, our first indication we are targeting. It's an very severe cancer. 1 out of 20 will be alive in 5 years only, and that's because it's very difficult to remove completely because it has very invasive growth into the brain, and for being able to remove everything, you need really to see deep into the tissue. The other aspect with the brain is that there are some critical functions in the brain. It could be the center of speech, it could be the movements, and you cannot just remove the entire brain. You will have to be very selective and very precise. So precise surgery in brain is, of course, very important for the surgeon. Additional purpose for us choosing is that the surgeon already are well trained and used to a high level of technology in the surgical room. They're also used to guidance by fluorescence, and I come back to that. And that's one aspect as well that makes penetration easier. Again, it's very important for us that all the things barriers that can be removed from us to go into the market quickly, we would like to remove those. So if I may have the next, please. So coming into our clinical trial and clinical development, this is the second reason why Fluoguide is a very attractive company seen from investor point of view. We have a very short and direct path to the market, and our clinical trials will be designed in the same way as I just shown before on the animal studies. So it will be done under white light. The surgeon will remove what they can, then the fluorescent light is switched on, and then possible more cancer is removed and sent to the pathologist to validate that it in fact is cancer that is being removed, and that what does not light up is not cancer. And that really, of course, is the endpoint of the study. So the positive predictive value is when the cancer or the tissue lights up and is validated by a pathologist that is in fact is cancer. And a negative predictive value is that tissue that does not light up in fact is validated not being cancer by the pathologist. And the positive predictive value is the endpoint on which we can get regulatory approval, and that will be the endpoint of our phase 3 program. And that again is very important because it takes us a few weeks for every patient. We are not looking at survival, for instance, or survival-free recurrence of the cancer or relapse of the cancer. So we really look at this positive predictive value, and that means that every patient that is included into the study will be in the study in 2 to 4 weeks, and then we have the result, and that's why we have a very short treatment of each patient. There will be 2 studies that would be needed for us to get approval. The first one is phase 1/2 study. That's the one we have ongoing for FG1, and that primarily look at safety and the optimal dose. Thereafter, we also get the proof of concept, and we get, last but not least, we get an idea of how much benefit can be added. I mean, one thing is that our product light up cancer. That's very nice for us to know, but really to know how much better is it in the hand of the surgeon, how much better is it compared to existing technologies. That really predicts the potential price we can get, and then again the value of the company. So it's very important that we get an understanding already now on how much benefit our product really adds to the community. And we don't want to go out just saying that our product can light up cancer. We really would like to go out adding a benefit to it, because that will add us the likelihood for us to get a good reimbursement, and then it will really give much better value for Fluoguide. But that was the first study that's ongoing right now. The second study is a confirmatory study, and that is looking at 2 things. We have to confirm efficacy and we have to confirm safety. So the efficacy can be confirmed on quite a few patients because the patients are their own control. So about 150 patients would be needed in a phase 3 program to document the effect. We probably need more in the safety, and we estimate that up to about 500 patients, but that can be pooled across different indications, and that's why it doesn't really matter so much that we have to have 500, because what we will do is that we will not only have a glioblastoma study ongoing, but also a couple of other indications on which we can pool the data for supporting safety. So the second thing here really is that we have a short and simple way to the market with quite a small clinical trial. So we may have the next. So status of our ongoing trial. What we're doing right now, as I said, I mean, we're looking at safety and we're looking at getting the optimal dose. And that is designed in a way that we have 3 patients per dose cohort. And that means that every time they are dosed, we evaluate the safety, because it's a phase 1 setting and patient safety, of course, very important. So we evaluate the safety, and if okay, we advance to the next level, and so forth until we have 8 cohorts in total. Right now we have had 5 cohorts through, and the product has been demonstrated to be well tolerated all along, and that's completely in line with our expectations from the preclinical program. In preclinical, we did a study where we look at 4 weeks toxicity, and we look at a program that will support approval of our compound, really. And we did that because it both looked at clearing UPAR used for guiding surgery, but also our product. So we had 2 kind of purposes with the program. And what was very nice with it is that we were stopped not by toxicity, but by feasibility. So we kept increasing the dose in the animals until we couldn't get more product given to the animals. And at that point in time, we had to stop, and that was more than 100 times more higher dose than what we really expect to end up needing in clinical trials. So that means that we have a huge therapeutic windows, and at the top we was not stopped by safety, but stopped by feasibility. So we did not expect to see any side effect, and that's also what have been the situation for us. Of course, you never know when you go into patient. So another thing, which of course is very important for us, is that we can see that FD1 lights up the cancer. And when the neurosurgeon is standing there and look at the cancer, I mean, she can clearly see the center of the cancer and can see that that lights up. Really, what is really important for us and what it really will make as I mentioned, the magnitude of benefit will be how precise can we see it in the border. And that will first be seen by the pathologist. So we take samples from our patient. All patients are sampled and is sent to a pathologist, and then from all of the patient, including into the program, will be read at the same time, blinded by a pathologist. And we need to do it that way because we cannot blind it for the neurosurgeon, so we can only blind it for the pathologist. And that's, of course, important for us that the slides are read blinded. And that will be done end of the first part of the study, and again, end of the final study. So when we, during the next few months here, we expect in Q3, have the optimal dose, and that's both in the level of dose, but also in the administration, if you give it in the morning or in the evening, also investigated right now. Then we lock the dose, and that will be the dose we live with for I would say, the rest of our life until we get it approved. And that's why it's very important that we select the right dose and the right administration. Then the part 2 of the study will be done exactly similar to the phase 3, just in a much smaller scale. So we have 2 centers, one in Denmark, one in Sweden. We use exactly the same endpoints we use in phase 3, the way of measuring it, everything will be exactly the same. And that is used then to do the power calculations, so we can go to the agency with a very good packages to say in the phase 2 meetings to get clear and buy into the clinical program we need for the phase 3, and not least, but at least also the number of patients needed for the phase 3 program. And then we'll have the final proof of concept. All that will come in Q3 for the safety, the first part of it, and Q4 for the second part of it, of this study. So that's all ongoing, very exciting at the moment. So we may have the next slide, please. So that's an overview of our pipeline. I mean, FD1, our lead indication is this aggressive brain cancer, glioblastoma, or high-grade glioma. It's the same, more or less the same thing. We have one prevalent indication we will start up with now after we have had the safety data for the first part of it. Then we go out of this strict phase 1 setting, and we go into a more, let's say, easy to do and a recruit patient. We don't have the same setup for the patient. We can do many patients at the same time. We don't have to take them one at a time. And that is what we do in Q3. We will submit at least one prevalent indication, probably long breast. Then we have the second product that we just licensed in last summer. And it has a slightly different profile than the first product. It's also an UPAR-targeted fluorophore, but it's better suited for stomach cancer because of the way it's excreted by the kidney than FD1 that is excreted by the liver. We have very strong preclinical data on FD2, and that is going into development next year. Then also recently we just acquired the Photothermal Therapy used for FD1, and that's a very interesting feature. So if we may have the next slide, please. So photothermal therapy is, I mean, as I mentioned before, the brain cancer is very difficult to remove precisely because if you imagine that you have the cancer invaded into the center of speech, the neurosurgeon will not just remove it because that will disable the patient so they cannot speak after the surgery. So they have a choice to make if they should leave cancer behind and then preserve critical functions for the patient, or that they should remove the part of the brain that contains the cancer and then create a disability for the patient post-surgery. And for instance, the center of speech will often remain, and about 30% of the cases, approximately 1/3 of the patient will have cancer left behind because the neurosurgeon cannot or do not want to remove all cancer of the patient. And here comes in photothermal therapy, because what really is a very unique feature with the FD1 is that when you light on it with a very strong light, you can actually heat up the fluorophore, and that means you heat it up to a temperature beyond what is damaging tissue, and that means that you can actually destroy the cells on which it's bind. And as you saw earlier in the presentation, it's binding very specifically to the cancer. In that way, you can destroy cancer cells very specifically. And more importantly, when you light on them, you can light on them more centimeters, 1 to 2 centimeters into the tissue, and that means that if you have normal tissue above or between the light source and the cancer cell, it will not be damaged because there are no FG1 bind to it, and it will not be heated up, and therefore you will only very specifically kill the cancer cells deep into the tissue. And that's the way to bring surgery to a cellular level, and we quite a lot of expectation for this let's say feature of FG1. So if I may have the next one, please. So competition. 2 products was actually approved some years ago, and one was from Photocure in Norway, it was Hexvix for bladder cancer, and the other one was Glioland for a Japanese German company for glioblastoma. Both of those products are based on visible light, and that means that you can only see it 1 to 2 millimeters into the tissue. Practical purpose, you can only see it on the surface of the cancer of the brain. Another feature of it is that the part of the product that lights up is a metabolite on the product that you administer, and that means that it will form a kind of cloud around the cancer and not specifically bound to it. And the last point is that both those 2 indications are very dependent on the administration. So for the bladder, you give the product into the bladder, and for the brain, you give it perorally, but you are depending on a non-intact blood-brain barrier. So that means that the 2 products are unspecific but still indication specific, and they are not. You cannot see them very deep into the tissue. But quite importantly, those 2 companies have started building this market, and really that's what is very important, and often for the medical technologies, it takes a long time to mature a new technology in a new market, and that is what's going on. So there's a question of timing when this market really takes off. Now there's coming in a range of different products that are, say, 2nd generation product. They're almost all based on a near-infrared light, and that means you can see it 1 to 2 centimeters into the tissue. They are all bind specifically to something and make them, let's say, less cloudy, but more specific in that sense. And 2 products of particular importance I will mention is one is for Untitled, and that is being submitted for approval in ovarian cancer, and the other one is for SurgiMap that had a phase 3 study ongoing for colorectal cancer. Some of the products also are very dependent on a specific equipment. That's one decision we did at the very beginning, that we did not want to be dependent on equipment. So we want to maintain this equipment independency because that gives us more partner flexibility and better penetration in our view. So the thing really that is unique with Fluoguide and FG1 is that the UPAR target is both cancer specific, so it binds only to cancer, but it's cancer type unspecific, so that means that they can use it for many different indications, and that's why we can go in on one rare indication, glioblastoma, get to the market quickly, and then spread out to the more prevalent indication. That's exactly what we want to do. So if we may have the next, please. So another thing that is very important with Fluoguide is that the technology evolution taking place in surgery. So for the last decades, a lot of more technology is coming into the surgical rooms, and that really helps the surgeon to become better to do what they already want to do. And examples are microscopes, it's robotic surgery, it's endoscopes. So a lot of the equipment is coming into it. And some of the kind of surgery are more equipment intensive than others. And for instance, the brain, because of microscopes has been used for a long time, is very technology concentrated. Lung cancer with endoscopes and also robot-assisted surgery are also becoming more technology intensive. But it's really a development that takes place over time. And I think that this, with the chart here, really is important, that this trend, that the digitalization, minimization, and also automation really help us in the sense that the equipment manufacturers need something to guide their equipment after, such as our FG1, but also the other Fluor developer companies, dye companies. But really, we are in a market where the trends are supporting us and where really all the stakeholders really like us and because they need us in the market. And that's a very important point for Fluoguide as well. We kind of talk with every company listed here on the slide on different levels. And on the brain side, the glioblastoma, we would expect that we have an help in going to the market kind of deal. It will not be something that gives us money to Fluoguide, but that will give us facilitation of going to the market quicker. In indications like lung, breast, and colorectal, the more prevalent indication, it's much more likely that we get a larger deal with a monetary element to Fluoguide. And that's also why we want to have more than one product to develop, because if we can, as an option, sell the entire product off to a partner and then still keep the other one for the other indications we are working with. So we might have the next one, please. So the team behind is, as mentioned, we are covering all the functions. We are not a big team in that sense that we don't want to be a lot of people, specifically not here in the beginning. We want to have a lean organization. We want to have low burn, a fixed burn rate, but we want to have the competences we needed. And here we all starts with Andreas Kjær, that is board-certified nuclear medicine chief physician for Rigshospitalet, and also have a professorship for Copenhagen University. There's a group of about 40 to 50 people working on his team on cancer imaging and diagnostics, and it's really where most of our products is coming from. They come from Andreas' laboratory, and we then license them from Copenhagen University and or buy them, as we did with FD1. And then we start developing. And the next one in line is Grete, taking care of all the production and the preclinical development. And that's something that many do not talk so much about, but it's where things often go sour when you do a mistake there, because then it gives delay, costs a lot of money, and it's really a nightmare. So for us, it was very important to have a super skilled person on that position, and that is Grete. She's done that before in Ascendis Pharma, US floated company, and a quite valuable company. And it's the same kind of complex molecules she's working with here as she did there. So the next in line is Dorte. She's both have experience from clinical development in device and drugs. And that's very important. We are under the drug regulation, but the device philosophy is important. And that's what Dorte is able to do. Henrik came in as a CFO end of last year, and has been instrumental in our issue we have just done now, but also as we become more focused on commercial deals with partners and so on, it's important that we have more commercial persons on board. Myself, I've done it before with Nanovi, where I took a product from the bench to the market, and it was a medical device, but an oncology company. And now I want to do that again with Fluoguide. So if we may have the next one. So we just have a directed issue, as I mentioned, on 75 million Swedish crowns. It was quite nice in a sense, not only for the money, but also because we had almost 15 new investors coming into the company, institutional investors, and half of them from Sweden, half of them from elsewhere internationally. And it's really several of them came in with an idea of further funding the company, assuming that we deliver what we have promised and what we will promise now. So that's really a very nice basis for and give us flexibility, robustness, and also validation, really. We was IPO'd in 2019. We had a very steep and very nice development. We had a best IPO of 2019. We had also grants. We had a 2.5 million European grant that we have half of it was paid out last year and the other half this year, next year. And then we have a very incentivized management and a good support from our two institutional investors that already were here before we actually did this directed issue that were linked and Arbejdernes Landsbank. And you can see that we have almost 9,500 shareholders. May I have the next one, please? So to sum up, I think, as I mentioned, Fluoguide is a very interesting investment opportunity, but particularly because of three things. There's a very huge market. A lot of patients have cancer. A lot of it comes back after surgery, and we have a very unique technology, UPAR illumination of the cancer. We have very interesting trends driving the surgical field. It's minimalization, it's digitalization, automation. And then we've already shown for our first product that it works and that it's safe, so we are well positioned to take care of it. We're no longer a binomial case, we're actually a case with a flaw, and now we have to establish how high can it be. We have several important milestones, safety Q3, proof of concept, magnitude of benefit Q4. We start a prevalent indication here later the year, and also 2022 will be very important, transforming Fluoguide into a late-stage clinical company where we initiate a phase 3 study in FD1. We have the FD2 will be started, and we have the photothermal therapy that will be started as well. So thank you very much for your attention. I'm looking forward to your questions.
Thank you very much, Morten, for an excellent presentation. You actually covered almost all of my questions, but I have some for you. I will take the first one from the web, and then someone is wondering, what can investors expect after the launch of FG1 in US and Europe? What can we expect the liquidity to result into?
What you can expect of the shares or the product?
I guess of the product, or what you want to build the company into long term, I guess.
Yes, okay.
Yeah.
No, I think that, I mean, of course, a company of our kind, we should always be open to exit. I mean, if somebody comes by and gives us an offer, we should sell the company, of course. But we would like that we do that not on the first occasion where somebody comes by. We want to do it on the best occasion. And to give us flexibility, I mean, that would be the best way really to get the best deal, is that we are not forced to do an exit soon, but we can do it when we feel is best for our shareholders. And for that purposes, our first product, FG1, is targeted glioblastoma. That's a very concentrated indication, and we have very few centers in, for instance, US, but also Europe, that takes care of this treatment, and it's an launch we can take care of ourselves. And then on the other side, we do the more prevalent indications, such as breast and lung, and that would more be the exit indication or the larger partnering indication, and that's where we can sell off an entire product if we want to, and that's why we want to have more product in the pipe. So to answer the question, we want to be a company that can take our first product to the market ourselves with a very small team and get a very nice revenue, but the real upside comes in from more prevalent indication coming later.
Yeah, very interesting. And you actually covered this a lot in your presentation, but this is very unique to your case, so I really want to know more about this. Usually it takes from, I think it's an average of 8 to 10 years for a company to reach from clinical stage into receiving a market approval, and you can do this in 4 years, or what is it?
Yeah, that's true. I mean,
yeah.
as I mentioned, we have quite the small clinical trials that is needed for us to get approval, because we have short treatment of each patient, and that we have the patient are their own control, so we don't need so many patients. So for that sake, we don't need so large clinical trials, and it's more a device-like case than really a drug-like case. But we are under the drug regulation, so yes, that's one of the benefits for Fluoguide, we believe as well. And we have been developing it quite fast. We was incorporated in 2018. I must say then there has been a lot of decades of research underneath, and that's what we established on. We don't have that in-house. We are doing it through agreements with the University of Copenhagen and also Rigshospitalet. But when we started out in 2018, I mean, from 2018 to now, and you can see from now then to 2024, a lot of things can happen.
Yeah, I understand. And why did you choose glioblastoma for the first indication?
Mm-hmm. Yeah, I mean, there was a number of reasons for it. One reason was that it's where the neurosurgeon are by far the most educated fluorescent guided surgery in the field. We have equipment that's well penetrated, so we can plug and play with our product. And we have a product, the 5-ALA, the Gliolan, that you cannot see so deep into the tissue, but still is selling in the field. So that means that we can have a kind of a reference case for approval. So that means that we do not have to, we can just compare to 5-ALA and show that you can see our product deeper into the tissue. And then we have a floor case in our you can say, in the investment case. And that we consider very nice. Then we build on top of that by adding more benefit to it. So that could be everything that reduce the likelihood of a neurooperation, less aggressive treatment, saving critical functions in the brain when doing the surgery, and then add to the price and the market. But we have a flaw in our investment case, and that's been very important for us to start with. All
right. And given that you show encouraging data in this glioblastoma indication, how big of a validation would this be for the rest of the indications you're planning on pursuing?
Yeah, no, it's a good question. I think that what happened during our study, which is actually pointing on that direction, is that one patient was included into our trial accidentally. And so when you do the glioblastoma, you're not always sure that it's glioblastoma. So some patient would be undergoing surgery, but not having glioblastoma. And this was actually a patient with a non-brain derived cancer. So it was an opportunity for us to test it in this indication, and it worked very well. Of course, it was only one patient, but it's underlying that the technology will work as well in other indications. And we have preclinical data on it. We have a lot of literature on UPAR, so there's very strong indirect evidence that will work. Of course, now this one patient is one step in that direction, and the final proof, of course, will come when we start the prevalent indication studies here after summer.
Okay. And just to understand the study design in the phase 3 and phase 1 and 2, of course, it's the same essentially. How does it depend on how good the surgeon will be in removing the tumor?
That's a very good point, and I think that is exactly the point that some of the companies are struggling with if they are not selecting a cancer indication with a reference product. Because a good surgeon will do better surgery than a poor surgeon, and that's FDA, I mean US Healthcare Authority. They would like you to see that you compensate for this bias of the skills of the surgeon. So by using a product of reference, and we use this gliolan as reference in our studies, then we actually remove the surgical expertise and skills, and that's actually a way of where we can reduce the number of patients we need and still do a very precise study. So it's a very good point, but it's particularly of a point if you do not have a reference, objective reference point.
Okay. And moving back to the broadening into different indications, do you see any potential for FG1 being used off-label once you get approved in the first indication, so you can have sort of a easier way to market in the other indications?
Yes, I mean that's of course a possibility, but I think that's more possibility in Europe than US, where they tend not to like it so much because of the liability. And I think also that I mean, we cannot, at least not actively promote it, and would not do that, of course, because that would be illegal. But I think that what is important as well is that, and that's a very conceptual thing, that we can choose to launch our product as just a tool for the surgeon, or we can choose to identify the benefit it adds to the surgeon or the surgery, and then document it by clinical trials, and then get a much better price, higher price for it by reimbursement codes and so on. And I think that the last one would be really important for us. That's what we are working on right now, to evaluate what would be the best investment case. Should we just make a replacement strategy on a lower price, quick launch on a gliolan, or should we go for demonstrating the benefit, proving it clinically, and then have a better price in a larger market. But I think it's worth considering doing this higher price, higher benefit launch. And if we do that for different indications, we have to do clinical trials for different indications, as the business case for the hospitals will be different for the different indications.
Okay, I understand. All right, Morten, time is running out here. It was very nice to have you here with us today.
Yes, thank you very much, Ludwig, and thank you, everyone.
Yeah, see you soon.
See you. Bye.